So this will be, I wouldn't say rather long, but it's one of the longer lecture bits. So we're going to talk about proteins and amino acids. Now, proteins in the body, they do pretty much, oh, sorry about that, yes. Proteins in the body, nothing important, trust me. In the body, they do pretty much everything, okay? If you think about any function that the body carries out, by function I mean, say, movement, okay? What happens in your muscles when they contract? Protein chains move next to each other. When you think about reactions that happen, for instance, in your gut during digestion, the enzymes that carry out those reactions are proteins. When you think about signals within the cell, the signals are carried out often by proteins. So by and large, proteins carry out 99%, I don't know, of the cellular functions in the body. And all the proteins are made from, well, strictly speaking, 21 different amino acids. But amino acid selenocysteine is relatively rare, but it's found in every person. So amino acids, those 20 different amino acids, when they form protein, they can do it in different order. And that order will determine the characteristics and functions of the protein itself. As we mentioned, protein molecules are one of the most important components of cartilage, hair, muscle, skin. They also function as enzymes, regulating cellular processes, and can transport and can serve as signaling molecules. So this is just multiple examples, okay? Please don't memorize, just understand the function. So structural, like collagen, contractile for muscles, transport, we just talked about lipoproteins and hemoglobin storage, casein, it's the protein storage in milk, actually great example, ferritin stores iron, hormones, a lot of hormones, pretty much all hormones that are produced by pituitary gland, not all of them, but most of them are proteins or peptides, enzymes, and part of your immune system, immunoglobulins, such as antibodies. So amino acids that comprise proteins, the molecular building blocks, have same and different parts of its structure. So first of all, there is a central carbon, also known as alpha carbon. This is sort of a, the merging point, okay? So alpha carbon carries on itself carboxylate group, which is acidic, and ammonium group, which is amino group. Okay, I'm going to change it to amino group. It's a basic. So the third bond is reserved for the R group, which is sometimes called a side chain. So in this ball and stick image of the amino acid alanine that you see here, you can clearly see carboxylate group. You can clearly see ammonium group, and you can clearly see the side chain, or R group. the structure. Does it make sense? So you may notice, we're going to start with this, you may notice that pretty much all amino acid share the same part of the molecule. So that part is the same in different amino acids. The side chain is different though. And it is the side chain that determines the amino acid properties. Amino acids are broadly classified into polar, such as asparagine. It's polar because of the amido amide group at the end of the side chain. And nonpolar, such as valine, which contains this, is isopropyl substituent, which is, hydrocarbon, so therefore nonpolar. So these are nonpolar side chains. Glycine is hydrogen, alanine has methyl group, valine has isopropyl, leucine isobutyl, isoleucine another sort of rendition of isobutyl, toluene group in phenylalanine. I don't even dare to name this. That's methionine. This interesting cyclic amino acid called proline, and tryptophan. So all these guys are nonpolar. Okay? Now you can see that the amino acids, the names, are often abbreviated. Okay? Usually first two letters, third may differ slightly, but in polar it's all going to be like, isoleucine, leucine, all EU. So there's some variations, but you can figure out what the acronym means. Okay? Now, they also can be encoded by single letters. Now, hear me out. I'm going to exemplify something. Glycine, G. Make sense? The first letter, right? Alanine. Make sense? Valine. Reasonable. Leucine. Isoleucine. Methionine. Proline. All make sense. Clear? And here we have phenylalanine. We can't use P because it's already taken. So instead we use the letter that mimics the sound. Phenylalanine, F. Now, you may wonder why tryptophan is W. Ran out of letters. T's been taken. Okay? Does that make sense? Now, within polar amino acid, you're going to find a wider, variety. There are more polar amino acids than nonpolar. So you can see the polar OH group here, here. The polar SH group. The polar amino combined with carbonyl, so amido groups. And here's, by the way, the illustration of what I was talking about. So you see tyrosine, the letter is Y, because T has been taken for threonine. Asparagine and glutamine, A and G are long taken, so N and Q. Now, these acids that you see, these amino acids, don't have a charge. They're polar, they don't have a charge. Now, there are so-called two acidic amino acids that have carboxylic groups at the end of the side chain. Okay? You see that these groups lost the hydrogen, so that would be at any other pH, they would look like this, but at the neutral pH, they donate hydrogen that get deprotonated, and this is the property of acids. This is why we call this amino acids acidic. So aspartate and glutamate. Now basic amino acids act as bases at in aqueous solutions at the physiological pH, forming NH3 plus and OH minus, or something similar. So you can see that all nitrogens in histidine, lysine, and arginine, all nitrogens are protonated because they act as bases, so therefore basic amino acids. Now, before we get to the structural formulas, you probably wonder if you need to memorize all the structures of all 20 amino acids. What are your thoughts? No. For the exam, I will, if you have a question about, you may get a question, identify the following amino acid as polar, non-polar, basic, or acidic. Clear? In this case, you will have the structure. Or, if the question is, which of the following amino acids is basic? Valine, lysine, serine, aspartate. You will have all the structures at the end of the exam. You can go to the very last page and check. Clear? Okay. So as we mentioned before, the alpha carbon, hydrogen, carboxylate, and ammonium groups are all the same among different amino acids. On the other hand, the R group differs. So this is how we draw an amino acid, obviously. So highlighted is the R group. Okay? The blue highlight shows you the R group, the part of amino acid that differentiates them. So now let's try to identify these amino acids as polar or non-polar. I am going to highlight the parts that are common. So remember, we're not looking at the common parts. We're looking at the side chain. So unfortunately, they did not do glycine justice. So I'm going to remove that and add hydrogen here. So glycine, any polarity within the side chain. This is the side chain. Just hydrogen. Any polarity? No. In threonine, when you look for polarity, look for oxygen or nitrogen atoms. Any polarity in the side chain. Those are polar bonds, so polar. So 3-letter, 1-letter abbreviation and drawing the structure. Folks, let's get in here. Okay, cysteine, I got it. Valine, where's valine? Yeah, so isopropyl. So this is how I usually draw it. For valine, look, you put the central carbon. Sounds good? You put the hydrogen. You put carboxylate group. You put amino group. Okay, you can do the same for cysteine. And then you draw side chains. So for valine, the side chain will look like this. Infocysteine, it will look like this. And just to make sure that, we repeat and that it sticks, this is a nonpolar side chain, and this is the polar. Side chain. Are we good so far? Now we're going to stop right here and continue Monday, because talking about protein structure, it's a long conversation. It's going to be a small chunk, but the idea is to maybe just start the conversation about levels of protein structure. So when two amino acids join, they form a peptide bond. You can see a peptide bond being highlighted with yellow. Okay? And when these two amino acids are joined by a bond, we call it a peptide. So when it's only two amino acids that are joined, we call it a dipeptide. 3 is tri, 4 is tetra. Now, what about 5? You can call it pentapeptide, you can call it hexapeptide. Usually we use the term oligopeptide. So oligo is the same as several. Okay? So the bond, the peptide bond, is considered to be an amide bond. You may remember we talked about the amides looking something like this. When you have a carboxylic acid and some kind of an amine, when you join them, what you get is this. So this is just a garden variety amide bond. Now, since amino acids have both groups, they have a carboxylic group and have an amino group, two amino acids can form that bond between them. And when we use the name, we change the name. Basically, here's the convention. Check this out. This is nitrogen. Agreed? So we call this N terminus. This is carbon. We call it C terminus. The naming is always from N to C. Is there any biological sort of rule towards that? To some extent, but for the most part, scientists just agreed on that. Okay? So the last amino acid on the C terminus does not change its name. In this case, it's alanine. And all other amino acids, they change the name by replacing INE, like glycine, into YL. Okay? So here, you have glycine. I'm going to make it larger, so it becomes glycel, and the alanine does not change its name. Also, that synthesis results in the production also of a molecule of water. We're good so far. Okay. So here's dipeptide. How about this? I'm just gonna do something here, some corrections. I'm not gonna redraw the whole thing. So we need to get rid of that. Okay? And we need to change. We need to remove 3. We need to remove plus. We're going to connect them. And we're going to remove that. That make sense? Clear? What I just did? Or you want me to do it again? Sure, yeah. This is what I'm asking. Okay. So basically your amide bond, the peptide bond, should look like this. The rest of the serine is going to be here. The rest of the threonine is going to be here, left and right. So what I did, now the only reason why I'm doing that is to avoid wasting time. Okay? So we already mentioned how you name them. You start at the N terminus and you change the name of the amino acid from INE to YL. Alanine, alanine. Glycine, glycol. The last one remains the same. Serine. Okay? So now, 3-letter abbreviations. I'm not asking to memorize this crap. Okay? I promise that on the exam 4 and the final exam, you're going to have the table with all the amino acids. The formulas, the 3-letter abbreviations, and 1-letter abbreviations. Make sense? You do need to understand, like, I'm not going to tell you if a certain amino acid is polar or non-polar or charged. That's on you. So 3-letter abbreviations. Let's go. So alanine, leucine, cysteine, methionine. Okay? Now the changes that you do, remember you change alanyl, leucyl, I believe it's this, cystyl. I was never sure about cysteine because, okay, and the same is here. Now, if you want, like, the only people that talk about peptides, the people who do biological chemistry and molecular biology. I've been in the field, well, I had been, not anymore, for a while. Nobody ever uses the full names. Ever. Okay? I can tell you how people would write down that peptide. It would look like this. That's it. Nobody even uses 3 letters. Everybody uses 1. Okay? Now, these 9 amino acids are essential, which means we have to receive them from food. Okay? Now, not every diet will provide all essential amino acids. So, if you are a meat eater, you're fine. Well, not necessarily even meat. Any animal protein contains all 20 amino acids, all that you need. Okay? If you do not consume animal protein at all, you have to provide yourself a combination of vegetables, grains, nuts, and beans. Does that make sense? So, an agreement is that if a polypeptide contains 50 or more amino acids, it's a protein. I personally disagree with that, this whole categorization, because I like to think, more about the function than this randomly assigned number of 50. What if it's 49? Is that not a protein? Okay? So, anyway, basically, proteins have several structural levels, and the first one is the primary structure. Primary structure is just a sequence. That's it. When somebody says that we alter the primary structure of a protein, means we alter a sequence. Okay? So why we pay so much attention? Because you can take, let's say, not a thyroid hormone that plays into my narrative that chemists should not play on the field of physiologists. It's not thyroid, it's the thyroid stimulating hormone. Okay? It does stimulate the release of thyroxine from the thyroid gland and has a very specific sequence of amino acids. And these amino acids are set in that specific order. Now you can imagine that you can change the order. Am I making sense? And if you change the order, the function will change. So the order is important. Which amino acid goes after which. Okay? So another example here is you have insulin here. So insulin is an interesting molecule because it has two chains, A and B, and they are connected by so-called disulfide bridges formed between individual residues of an amino acid cysteine. That is indeed the first protein which primary structure was determined. Moreover, it was the first protein. I believe during that work, insulin was not just the structure was determined, it was synthesized back, and it was shown that synthetic insulin has the same properties as the one isolated from pigs usually. Okay, does that make sense? Good. So, folks, I'm not going to ask you to scroll hastily through the table that contains all amino acids. Instead, I'm going to highlight amino acids that are on the N-terminus and on the C-terminus. So let's start with the N-terminus. N-terminus on that picture, on the left or on the right? So this is the N-terminal amino acid. Okay? And this amino acid is phenylalanine. Imagine you have that question on the exam. How do you know which amino acid is that? You go to the table that I give you. Clear? Cool. C-terminus. That. This is alanine. Okay? Name of the tripeptide. Where do you start when you name? On the N or on the C? Always. N. So, phenylalanyl. I believe it's cysteine, but I'm not entirely sure. Cysteine sounds better. Okay? Phenylalanyl, cysteine, alanine. And I don't really know why we're talking about that in the chemistry class, but I guess we must oblige, since I'm not designing the course. Yeah, we have a bunch of different peptides in our body, okay? Enkephalins. Oh, for God's sakes. No. Endorphins are not really polypeptides. Endorphins are molecules that are structurally similar to natural opioids, or vice versa. Natural opioids bind to the same receptors. Okay? So yes, enkephalins are polypeptides. Okay? A bunch of hormones that are produced by humans are peptides. I can actually give you a full, if anyone is going to take ANP2, I guess. It's very easy to remember which hormones are polypeptides and proteins by remembering which ones are not. All steroid hormones are not, thyroid hormones are not, and the hormones of adrenal medulla, like epinephrine, norepinephrine, are not. Everything else is either peptide or a protein. Okay, how about, we're not going to write the names. I just want to make sure that we all understand how combinations work. Okay? So, glycine is GLY, alanine is ALA. So, how can you, and letter-wise it's G, it's A. So remember, we're reading from left to right. Okay? Can we do this? Yeah. Glycyl, glycyl, alanine. Can we do this? Glycyl, why? Look. All tripeptides. We can combine them that way, right? Can we do this? Yeah, we combine them in a different way. Same idea, 2 glycines, 1 alanine. Do you understand what I'm trying to do? No? Okay. So remember, we have N-terminus and C-terminus. So just as an example, I'm going to draw this tripeptide. That's your first glycine, okay? This is your second glycine, so okay? And this is your alanine. Clear? So we read it from left to right, from N to C, GGA. So this is N-terminus, this is C-terminus. We can switch and do GAG, or we can put alanine first on the N-terminus and the rest is glycines. So the point of this exercise, well, in naming, obviously, the first one will be glycyl, glycyl, alanine, second will be glycyl, alanyl, glycine, and the third, alanyl, glycyl, glycine. Nobody uses that. I feel utterly stupid even saying those things. Nobody ever talks like this in biochemistry. It will be just letters. Okay? The point is, those 3 different tripeptides are going to have different properties. Well, we're going to pause right there. So depending on the primary structure of the protein, the polypeptide chain will assume other conformations, assume a certain shape, which can be characterized as the secondary, tertiary, and quaternary structures. So here we will try to establish what exactly those levels of the protein structure are. So in the secondary structure contains 2 major types. One of them is alpha helix. So what it's formed when the hydrogen bonds between the partially negatively charged oxygen and carbonyl group and partially positively charged hydrogen of the amino group form these hydrogen bonds, and these groups are located in two different turns of the helix. Here you can see that this hydrogen is located in one turn, and this oxygen is located in another. This hydrogen is located in one turn, this oxygen is located in another. When we were talking about bonds, I constantly used the phrase that the strength is in numbers. Hydrogen bonds are relatively weak, but the formation of multiple such bonds stabilize the helical shape of this fragment of the protein. Another type of the secondary structure is also stabilized by hydrogen bonds, and it is called beta-pleated sheet. So, same story, partially negative oxygens of carbonyl groups and partially positive hydrogens of the amino groups form hydrogen bonds. However, this time these groups are located not in different turns, but in different layers of the sheet. So for instance, this carbonyl group is located in one chain, and this amino group is located in another. Now you can see that this beta-pleated sheet is sort of, these strands are anti-parallel because the direction dictates, the direction is dictated by the interactions between those groups. Some proteins can actually form a triple helix, which is not intramolecular, but rather intermolecular structure. So here you can see that there are 3 polypeptide chains that weave around each other, forming this triple helix. Very much like, the hair woven in braids sometimes. The functional importance of this arrangement is that these triple chains that, remember, are held by hydrogen bonds, again the strength is in numbers, provide extra strength for the protein collagen, which is the main protein fiber type in tissues like skin and tendons and ligaments. In other words, in all connective tissues. So in some cases, the abnormalities within the secondary structure can explain certain pathological states. For example, in the disease that you absolutely know, called Alzheimer's disease, beta amyloid proteins normally exist in a form of the alpha helix. However, in Alzheimer's, due to the changes in the structure of the beta amyloid proteins, they change shape from alpha helices that are soluble within the cytoplasm to beta sheets, and that results in the formation of beta amyloid plaques, basically insoluble proteins. In addition to changes in beta amyloid, another contributor to the Alzheimer's disease are so-called neurofibrillary neurofibrillary tangles, or which formed by so-called tau protein. So this is the illustration of what is going on. The normal beta amyloids are alpha helices, and beta amyloids in Alzheimer's are beta pleated sheet. Now, secondary structure of the protein can actually, we used to be able to predict it fairly reliably by analyzing the sequence. But this secondary elements, first of all, not the entire protein is folded into either alpha helices or beta sheets. There are some parts of the protein that do not form any secondary structure. Eventually, all the elements of the secondary structure, whether they are defined or not, fold onto each other, forming a tertiary structure. So this is the overall shape of the folded polypeptide chain. So this shape is determined by the interaction between side chains of various amino acids, which results in bending and twisting of the shape. Now, why? I don't like the question why, but why is a certain protein, why does it acquire a specific shape? The answer is, FYI, not for the exam, the answer is the energy. Every molecule can be characterized by free energy. We briefly talked about it. Every molecule structures to achieve the lowest free energy. That is what drives it. So, again, the determinants, attractions and repulsions and other interactions between side chains, and sulfur disulfide bonds, the crosslinks. Now, let's take a look at the tertiary structure of that protein of the unknown type, but it allows us to illustrate which interactions contribute to the formation of the tertiary part of it. So first of all, salt bridges. Amino acids such as lysine contain positively charged amino group. Amino acids such as aspartic or glutamic acid contain negatively charged carboxyl group. Positive and negative charges form a salt bridge. Polar amino acids, let's presume that these are threonians. I believe it's a threonian, which contain polar hydroxide group, for examples. They can form hydrogen bond between slightly positive hydrogen of one amino acid and slightly negative oxygen of another. Then another example of hydrogen bond. Then there are hydrophobic interactions. For example, these two molecules of residues of phenylalanine or these two residues of alanine can interact with each other via basically the dispersion forces. Now, this actually begs the question to some extent, which groups will be exposed on the surface of the protein and which groups will be buried in the depth of it. So to answer this question, I will ask the question first. What is the most common chemical in the human body?
Water.
It's water, right? Everything in the human body, every cell is a bunch of water, basically, right? So these proteins exist in water. Now hear me out. If you have something that is surrounded by water, do you want the hydrophilic parts of that molecule to be exposed towards or away from water? Dude. Water is around you. Do you want to show water that you love it? Yeah. So, the rule of thumb, the surface of the proteins is hydrophilic. Hydrophilic elements will be on the outside. Hydrophobic will be hidden on the inside. So this provides you a list of those interactions. Hydrophobic, hydrophilic, such as interactions between the water and polar side chains, salt bridges between the charged, oppositely charged side chains, hydrogen bonds between the polar side chains. And something that I completely forgot to mention, disulfide bridges here. These are disulfide bonds that covalently, and I can't emphasize it enough, covalently connect to chains. So while you can disrupt some of those interactions by, let's say, changing the pH or adding salt, disrupting disulfide bridges requires an actual chemical reaction. So now let's match all the letters, all the terms. So polypeptide chains held side by side. I want to illustrate this. We're talking about this. What do we call it? Flat. If it's flat, side by side, bends. Nobody wears these pants anymore. You know, like pleated pants? Yeah? Folded. So, folded, pleated. So A is going to be a beta pleated sheet. A sequence of amino acids. Which level of structure does it represent? Just the sequence. Come on, fellas. Remember, sequence is the primary. Now, I know that it's going to be on the record, but listen. I know this shit. I'm asking you to participate because it helps you learning. If you screw up, not a problem. Okay? Corkscrew shape with hydrogen bond stabilizing it. Corkscrew. How does it look? Alpha helices, yes, excellent. And 3 peptide chains woven around each other. There's no other option besides D. Triple helix. So, I'm going to give you some details about amino acids and you're going to tell me the interaction. Leucine and valine both are nonpolar. What type of interaction will they establish? If they're nonpolar. Exactly. 2 cysteines. That's the side chain of a cysteine. What would they form? 2 sulfurs. Disulfide bridge. Aspartic acid and lysine. These are charged guys. What do you think? What do you think? Yes. And hydrogen bonds? Obviously serine and threonine, because serine has OH group and threonine has OH group. Give me a second, let me shut the door. So, quaternary structure, you have several polypeptides that are shaped some way, and the several polypeptides, often called subunits, form the overall structure of the protein. Now, I mentioned that before, you should not allow general chemists to the, in the sniper rifle shot range of biochemistry and physiology. So what does that mean, that quaternary structure consists of 4 polypeptide chain? That is absolute crap. So quaternary structure is a combination of 2 or more protein units, period. Can be 2, can be 4, can be 5, can be more than that. There are some complexes that have more. So the point is that often for the protein to, perform its function, it has to have those 4 subunits. A good example of this is hemoglobin, which contains 2 alpha subunits and 2 beta subunits. So what holds those subunits together? Absolutely the same set of interactions that holds the tertiary structure together, meaning you're going to have ionic interactions, you're going to have disulfide bridges, hydrogen bonds, hydrophobic interactions, and so on and so forth. So this picture summarizes the structural levels, the sequence, alpha helices and beta sheets representing secondary structure, tertiary structure being stabilized by the side chains of amino acids, and quaternary structure, which is the assembly of several protein subunits. So the example when the replacement of one amino acid goes sour is the sickle cell anemia. So here you can see the comparison between the normal beta chain of hemoglobin and beta chain with a mutation. So glutamate or glutamic acid is polar, and it is conceivable that this polar amino acid, remember what we were talking about regarding the surface, right? Polar amino acids are on the outside, okay? So it is replaced by valine, which is non-polar amino acid, which implies that this particular part of the protein will try to hide from watery environment everywhere else, and shape of the hemoglobin will be altered. So what effectively happens is that the globular hemoglobin, this globular molecule right here, changes its shape to more like a fiber. It starts aggregating, and that results in the change of the shape of red blood cells from round to crescent shape like a sickle. That leads to obstruction of capillaries, inflammation, hypoxia, hemolysis, and kidney damage. So you can see what happens when the hemoglobin molecules acquire that mutation. They go from soluble 4 subunit structures to insoluble fibers, and that completely screws up the red blood cells. Summary, I'm not going to focus on the table, it's just for your reference. We're going to jump to matching these. So beta pleated sheet refers to which level of the protein structure? Alpha helices and beta sheets are what? Secondary. Order of amino acids then? Go on. Primary, yeah. Protein with 2 or more peptide chains, huh? Quaternary. Because when we start talking about multiple polypeptides, that's going to be quaternary. And the structure that is stabilized by the bonds between the R groups is tertiary. So since proteins assume a specific shape, that shape can be disrupted. We call this disruption denaturation. Now, hear me out. It can involve any level. Denaturation may impact only quaternary structure. It may impact tertiary and quaternary. And it may impact tertiary, quaternary, and secondary structure. What can cause it? Heat and organic compounds, for example. I'm trying to think about some good examples. Okay, I can give you a few examples. So heat, that question will be directed, I think, mostly to the female part of the audience. How can you make your hair curly? Heat. Yeah. What you do, you denature protein keratin that forms most of your hair. And when you denature it, it assumes new shape. What happens to your curly hair if you don't do anything, just let it go? Curls, they go away, right? Because keratin reassumes its natural shape. And the same is true for people with curly hair that straighten them up. Okay? Organic compounds. This is a very, very specific example. Don't ask me. But imagine, let's say you drink coffee with half and half, or milk. Okay? Good, so you add some milk. And then immediately you decide, I'm going to add a little kick to it. And you add a shot of whiskey, alcohol. You know what's going to happen to that milk? It's going to turn into curd. Because casein will denature in the environment that contains now alcohol. Acids and bases. Yes. Now hear me out. Are you familiar with the egg drop soup? Yeah? It's acidic. Not acidic to the extent that your teeth are falling out, right? But it does have an acid component in it. And when you drop an egg into an acid, it denatures and becomes kind of solid-ish. Okay? That's the idea. Heavy metal ions can denature. Agitation. Yes. So like when you whip the cream. I personally do not use that example at all for very silly reason. Cooking eggs as an example of denaturation is used in every textbook that mentions denaturation. I want to be a little bit more creative and also provide you an example of renaturation. Because when protein is denatured, when you cook an egg, it's not going to go back to its liquid shape. Like, it's done. Okay? But the example with the hair can renature. Okay, this is the summary of how different denaturation conditions can disrupt protein structures, in which interactions, so like hydrogen bonds and hydrophobic interactions, heat, hydrogen bonds and salt bridges, acids, bases, organic compounds, hydrophobic interactions, heavy metals, disulfide bonds, and agitation, hydrogen bonds, and hydrophobic interactions. So the answer, yeah, both are denaturation events. Because tannic acid denatures proteins that are released from the burn, okay, and egg, same story, denatured. So now we're going to talk about enzymes. These are biological catalysts, and to think about enzymes, first let's recall something here. Okay, so this is the energy profile of the reaction between carbon dioxide and water, which results in the formation of bicarbonate ion and hydrogen ion. You can see that without enzyme, the activation energy is really high. With an enzyme, activation energy is relatively low. That makes me asking you a question. Which parameter of the reaction is associated with activation energy? Let me rephrase it. When activation energy goes down, how does the reaction rate change? Think about this. Remember the hump analogy that I used back then? In order for the reaction to proceed, it has to go through the hump, that is activation energy. If the hump is lower, will reaction go faster or slower? Low hump. You have to go over the hill, right? Over which hill you're going to go faster? Hill that is tiny or hill that is really, really tall? The tiny one, right? Does that make sense? No? No? Okay. So, follow my black dot. So this is my black dot following the path of the reaction with no catalyst. It has to climb a really steep hill for a long time. This is the path with the catalyst. Not so steep, it will get to the end point much faster. Does that make sense now? So we can say that lower activation energy, increased rate. All good? Okay. So enzymes, just like any catalysts, can lower the activation energy and increase the rate of the reaction. Okay? For example, carbonic anhydrase is essential for the cells to produce bicarbonate, anion and hydrogen cation. Okay, enzyme names. First of all, this is chemists, not enzymologists. Enzymes have common names. A common name loosely describes what the enzyme does, loosely describes, and adds the suffix ase. Also, there is a formal official classification of enzymes in which there are no names. There are numbers, and it's like 14.5, 14.5.3. Whatever. I don't remember the exact. The point is, by looking at these numbers and go into the catalog, you can see what enzyme this is, but we don't use these numbers. So let's take a look. For instance, the enzyme sucrase breaks down sugar, sucrose. Then we have enzyme oxidase. Oxidase can oxidize a lot of different compounds. For instance, cytochrome C oxidase. Okay, or NADPH oxidase. Okay. Sometimes the names are common. Pepsin, trypsin, chymotrypsin. It's all are common names of the enzymes that are proteases. Do you want to learn the funniest enzyme names? There are two. First, those are actual enzymes. First, there's an enzyme. Remember, we're talking about membranes, biological membranes, phospholipid bilayer. There's an enzyme that moves phospholipids from one layer of bilayer to another by flipping them. The name of the enzyme is flippase. And then there is an enzyme that's produced by flu virus, which steals a certain chemical structure from the cell in the process called cap snatching. The enzyme name is snatchase because it snatches. Now these are 6 main classes of the enzymes. Oxidoreductases facilitate oxidation and reduction. Transferases transfer groups of atoms from one molecule to another. Hydrolases facilitate hydrolysis. Lyases, they either add atoms to the double bond or remove atoms from the double bond. Isomerases simply rearrange the atoms in the molecule, and ligases use the energy stored in molecules of ATP to combine other molecules. Let's take a look at some reactions. So, let's start from the bottom, because when you have a question like this, you want to eliminate answers that you know for sure. So, simplest, hydrolysis reaction. Which enzyme do you think will facilitate hydrolysis? Hydrolase. Yes, the first one. Yes, I got you. Transfer a group. Transferase. Yes. Rearranging atoms in a molecule to form an isomer. Isomerase. And ligase forms bonds. So how does that work? First of all, an enzymatic reaction can be, in general, check this out, described this way. So you have a molecule that you start with, which is called a substrate. And substrate is converted into a product by the enzyme. So the example that I can give you on this, let's say you have sucrose, and the enzyme sucrase will break it down into the glucose and fructose. So in order for the substrate to be converted to a product, the substrate is to bind to a special pocket in the region, in the protein, called an active site. Now, usually you can find very specific side chains of amino acids in the active site, which have the capacity to bind the substrate. Once the reaction is complete, active site will release the products of the reaction, and it means that the enzyme is not used, wasted, or modified in the reaction. Okay? Does that make sense? So here is the substrate bound into an active site. And if you think about sorry, I'm just trying to correct this. So if you think about the whole process of the enzymatic reaction, the first step actually is to form enzyme substrate complex. And this process, binding in, is reversible. Substrate can bind and then can leave active site. Once it is converted into product, then there is no way back. So you see that the first step is reversible and the second step is not. Okay? So what's important is that the enzyme is used over and over. And another important point that I want to make: enzymes do not change the reaction outcomes. Meaning that the ratio of products stays the same, but they do increase the reaction rate. So this example shows you what happens when the enzyme called lactase binds lactose in the reversible manner, and then the glycosidic bond of lactose is in a position so that side chains, the R groups of the amino acids in the active site, facilitate the breakdown of that glycosidic bond. Since the shape of individual molecules of glucose and galactose is different from the lactose, they can't attach to the active site anymore and they leave. In the meantime, lactase can react with another molecule of lactose. So the first model that described the enzymatic reaction was so-called lock and key model. In that model, scientists presumed that the active site was rigid and not flexible. And that meant that only substrates that can fit exactly into the active site could bind the enzyme. This model turned out to be incorrect because the tertiary shape of the protein is flexible. So how do we take that into account? How do we take into account the fact that the active site can adjust? So the answer to this question was provided by the induced fit model. In this model, the paradigm shifted. Enzyme structure is flexible, and it can adjust to the shape of the, hold on a second, I need to correct this, can adjust the shape of the active site in order to bind the substrate. Which means that each enzyme can bind a range of substrates. Which means we can design a molecule that will bind into the active site and block the enzyme from working. Shape changes allow to improve the catalytic functions. And this is the induced fit model. If you look at the induced fit model description of how lactase works, there are two pockets in the active site. One for galactose, one for glucose part of the lactose molecule. But when they bind, the shape of these pockets changes. So that shape change, that change in fit, was induced by the substrate. And effectively, this is the interaction between the substrate and the enzyme that is essential to acquire the proper arrangement to lower the activation energy. So one of the interesting features of enzymes is that they can exist in the form of isoenzymes, types of the same molecule that are found in different cells or the tissues of the body. So usually these enzymes have a quaternary structure which has slight variations. So different subunits can be in the different ratio to each other. For example, the enzyme called lactate dehydrogenase, LDH, that facilitates the conversion of the lactate molecule to pyruvate can exist in different combinations of the polypeptide subunits named M and H. For example, lactate dehydrogenase found in liver and muscle contains 4 M subunits. Same enzyme but in the heart, it contains 4 H subunits. So how is this used as a diagnostic tool? When somebody has a heart attack, known as myocardial infarction, can be diagnosed by levels of enzymes such as creatine kinase, found in the heart, and lactate dehydrogenase, specifically found in the heart, H4, which allows doctors to distinguish the heart attack from, let's say, a liver damage. Or even that, anyone watched House MD TV show? Come on, folks, I'm old, but not that old. It's like early 2000s. Watch it, it's great. Okay, and I think it's on Amazon Prime or Netflix. Like seriously, it's a good show about a hospital. Then my example becomes moot. Basically, the main character at some point goes through the infarction, but it's not in the heart, it's in the muscle. And you can distinguish the infarction, same idea, blockage of blood flow in the muscle versus the heart, by analyzing LDH levels, specifically whether it's M4 that comes from the heart, comes from the muscle, or H4 that comes from the heart. So this shows you different combinations of different subunits in various tissues of the body. I'm not going to dive into it, but it kind of supports a point. So let's answer these questions. The active site. The whole enzyme, the substrate, or a section? Remember, it's a part of the enzyme. Induced fit model. The shape of the enzyme after substrate binds. Is it the same or is it different? What do you think? Induced fit. Same or different? As you wish. Different, yeah. So it adapts to the shape. So enzymes aren't active, aren't have the same activity at all conditions. It can be affected by various external factors. All enzymes have an optimal temperature. Human enzymes, the optimal temperature is 37. Does it make sense to you? Now, this statement I have a problem with. Show little activity at low temperatures. There are organisms that grow exclusively at cool temperatures, such as 0 degrees Celsius. Their enzymes are definitely active at low temperatures. Okay? Now, human enzyme will lose activity at the temperatures that are high because they're going to get denatured. But there are organisms that exist in very high temperature environment, for instance, hot springs in Yellowstone. Those enzymes have optimal temperatures around 90 degrees Celsius. Okay? But the point is, each enzyme has its own optimal temperature that corresponds to the maximum activity. So here's my question. If you drop the temperature to, say, this, at this temperature, will enzyme be active? I'm not saying totally. Will it be active? Will it do something? Yeah. It's not going to be maximum, but turns out often we don't need maximum. pH also affects different enzymes have different optimum pH. Here's an example. We have an enzyme called pepsin that breaks down proteins in your stomach. Its optimal pH is 3 because that's the pH of your stomach. And then you have the enzyme called trypsin that breaks down proteins in your intestine where pH is around 8. Its optimal pH is between 7 and 8. The reason why low or high pH can disrupt enzyme activity is because it denatures it. It affects the tertiary structure. So I'm not going to read this table. Just shows you different pH for different enzymes. So if the optimum pH sucrase is 6.2 and the optimal temperature is 37 Celsius, how will the change of pH to 4 affect the activity of the enzyme? Will it increase it or decrease it? If you step out of the optimal, remember folks, it's always a decrease. Look, this graph shows you this is the decrease. So enzymes can be inhibited. if enzyme, so this is the normal, enzyme interacts with the substrate forming enzyme-substrate complex. If substrate is replaced by the inhibitor, the reaction does not proceed. So there are different types of the inhibitors, but by and large, what they do, somehow they prevent substrates from getting into the active site. Two types of inhibition that we're going to talk about, the competitive and non-competitive. So when inhibition is competitive, the inhibitor usually has a structure that is similar to the structure of the substrate, and it effectively competes with the substrate for the active site. So let me illustrate something for you. Imagine that this chair is the active site of the enzyme. Okay? Jade is a substrate. She wants to get in that chair. I am a competitive inhibitor. I also want to get in this chair. Right? So we are competing with each other. How can a cell, which Jade represents, how can it basically offset the effects of me, the inhibitor? If you increase substrate concentration, in other words, imagine that Jade is not alone. There are like 50 Jades all near the substrate, all near the active site. Does that make sense? Do I have a chance? 50 Jades versus me? Probably not. Like, they're gonna, like, it's like a music chair with one chair, right? That's it. That's it. Okay? So, no way. So basically, if you look at this representation, when the substrate concentrations are high, the reaction is likely to proceed. When the concentration of inhibitor is high, the reaction will not. Okay, so this illustration shows you how the inhibitor prevents the substrate from getting into the active site. I want to give you a clinically relevant idea here. Is anyone familiar with the drug called Bactrim? Yeah? Bactrim is a mix of two competitive inhibitors with a single metabolic pathway in bacteria, sulfamethoxazole and trimethoprim. Okay? Another example is your garden variety penicillins that prevents an enzyme called transpeptidase, also a competitive inhibitor. Okay, binds to the active site. There are also non-competitive inhibitors. So these guys are very interesting. So non-competitive inhibitors do not compete for the active site. Instead, they bind to the site other than the active, so-called allosteric site. Allo means different, steric site, and the shape of the enzyme, shape of the active site, is now changed. So in our example, this is what's going to happen. So I am now a non-competitive inhibitor, and when I'm binding to the enzyme, the shape of the active site is changed. Okay? Jade cannot sit on that chair, period, as it is now, right? So it doesn't matter how many Jades are surrounding the place. it's not going to work anyway. Okay? Sorry, I cannot provide you with example of a drug that is a non-competitive inhibitor. I'm positive there's one, maybe more than one. I just can't get any examples from top of my head. Okay? Non-competitive inhibition is pretty common in regulating metabolic pathways in the cell. Here you can see in this illustration that the shape of the active site has been changed. The irreversible inhibition I am not sure that they are correct, because as far as I know, penicillin is not a reversible inhibitor. It's a competitive inhibitor of the active site. Anyway, so the idea of irreversible inhibitor is that this inhibitor somehow covalently modifies the protein, which effectively eliminates the enzyme activities. And I want to emphasize, hear me out, I am not comfortable with that statement. Kind of know it, but if you destroy an enzyme, at this point I don't think we can qualify it as an inhibition. It's just a destruction. Inhibition implies that the inhibited molecule otherwise remains, if you remove the inhibitor, it remains still functional. Okay? So obviously this table summarizes. I'm not going to read it to you. So let's describe each inhibitor as competitive or non-competitive. So if you increase the amount of substrate and it reverses inhibition, competitive or non-competitive? Competitive. More jades competing for the same chair. I apologize for using you as an example, but enzyme surface, but not the active site. Not the active site. Competitive or none? Structure is similar to the structure of the substrate. Competitive. I actually found a better example for non-competitive inhibition. It's not great, but imagine that instead of me being here, sitting here, there's a bear. Would you want to get any closer to the bear? I presume not. You see what I'm saying? So I'm somewhere else, but and inhibition is not reversed by adding more substrate. It doesn't matter how many jades. If I'm a bear, I don't think that 50 jades would still, matter. So non-competitive. So that concludes our conversation about enzymes, which is good.